Hi, my name is Kelly. I'm from the Center of Academic Achievement, and this is a biology clip that I just put together. Eukaryotic cell cycles are crucial for the overall cell and the maintenance of an organism. There are three purposes of cellular division in eukaryotes: reproduction, growth, renewal, and repair. Reproduction is handled by meiosis, whereas growth and renewal and repair are dealt with by mitosis. So today we're just talking about the mitosis section of the cell cycle. What you see here is the textbook example of the eukaryotic cell cycle. It starts with G1, moves to S, G2, M, and C. During G1, this is the time where the cell is growing, producing proteins, and just basically living out its existence. During synthesis, DNA replication takes place. During G2, there's a centriole replication. So G2 is basically the starting of mitosis, the M phase. So G1, S, and G2 all make up interphase. And like I said, during G1, there's cell growth and organelle duplication. And there's also protein synthesis that allows for cellular differentiation, which makes an eye cell versus a hair cell. They both contain the same DNA, but certain proteins are produced by each cell to differentiate it from another. During the S phase, there is DNA replication as well as histone synthesis, and DNA strands will unwind during this time. And then in G2 phase, the centriole replication takes place, and then there's a finishing of the protein synthesis that took place in G1 and a little bit in S phase. Then we get to mitosis, the M phase. This is broken down into 5 specific parts: prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, so that's the typical chromosomes that you normally see in a textbook. Spindle apparatus is assembled as a cytoskeleton breaks down, and then the Golgi and ER are dispersed while the nuclear envelope breaks down. So all of this is happening at one time, and even though this is separated into specific phases, everything is going on at the same time. So prometaphase, the chromosomes are attaching to microtubules at the kinetochore, so at the centers, and they move to the equator of the cell. In metaphase, chromosomes are aligned at the metaphase plate and are under tension, so all of these chromosomes are getting ready to be pulled to different sides of the cell. During anaphase, proteins holding sister chromatids at the centromere are degraded, and so individual chromosomes are pulled to the opposite poles and spindle poles are moved apart. So this is the part where the chromosomes are broken in half and the sisters will go to each side. And telophase is when chromosomes group at opposite poles and decondense, and this is when the chromosomes are no longer visible. They are no longer packed together, so they're getting ready to go back into interphase where they can start producing more proteins. The nuclear envelope reforms around these chromosomes, and the Golgi and ER reform. Okay, so this is the cell cycle as it's shown in a textbook. It's the same every single time. Things can happen, though, to disrupt this schedule. That's why we have certain checkpoints within the cell cycle. So there's a checkpoint at G1S. It's the start or restriction area. So once the proteins are made, once the cell is getting ready to start producing more DNA, this is the part where if the cell isn't healthy, it won't divide if it's working properly. At the G2M checkpoint, this is when the cell commits to mitosis. All of the DNA has been replicated, all of the centrioles have been replicated, all of the organelles are replicated, so there's enough to go in each of the new daughter cells. And then the final checkpoint is at the spindle checkpoint. And this is basically raising the question, are the chromosomes attached to the spindles? Otherwise, an unequal amount of chromosomes will be pulled into the new cells. So It's common to have failures in the cell cycle control. When this happens, cancer takes place. There are two types of genes that contribute to cancer. There are tumor-suppressor genes and proto-oncogenes. Tumor-suppressor genes normally inhibit the growth of cells which contain mutations, so they allow for apoptosis or programmed cell death. They play a large role in the cell cycle checkpoint, so if the cell is not healthy or it doesn't have all of the DNA or all of the proteins that it needs to get to that next point, then the cell will automatically die. It will go into apoptosis. There are a couple of examples that are classically given. The RB protein and the P53 protein. RB protein is mutated in 40% of all cancers, and P53 is mutated in 50% of all cancers. So this plays a huge role in getting rid of or eradicating cancer altogether. Proto-oncogenes are genes that also contribute to cancer. Normally, they can encode for receptors for cell growth or signal transduction proteins. These become oncogenes when they're mutated. So an oncogene will have the capability of causing cancer when presented to a cell. Examples include the RAS protein, present in 20 to 30% of all cancers, and the SRC kinase, present in 2 to 3% of all cancers. As far as the cell cycle is concerned, the biggest part to remember are all the steps that take place from interphase to mitosis. The overall take-home message is that mitosis and the cell cycle in general is a very organized, planned out cycle that, when interrupted, can cause havoc on the cell and on the organism. And it's important to understand mitosis in order to later understand meiosis and other genetic contributions that take place in the cell. Thank you. I hope this helped, and check out our other videos on the CFAA. Thanks.